Very good day, doctor. My name is Gajalakshmi Raja and my metric card number is S55261. The topic of my research is Gellan gum with stingless bee honey for dressing product. And my supervisor is Professor Khairul Anwar Mat Amin. What is Gellan gum and how does it work? Gellan gum is a nature-based ingredient which was discovered in the 70s. It was found growing in a pond in Transylvania. And it is a byproduct created by certain bacteria. And this byproduct can be duplicated through fermentation. And the unique abilities of Gellan gum: it keeps food and beverage product from settling and separating. It is helpful in creating vegan-friendly products such as plant-based milks, yogurts, and cheese. What makes it suitable for dressing product? Well, there's a list of factors. Let me list one by one. The first factor is it is able to generate a hard transparent gel. It is able to generate elastic and soft gels. It possesses high thermal resistance. It is a material with low dose. It has acid stability. And it has lower sol-gel transition temperature. It has high gel strength, highly biocompatible, and last but not least, it has ability to incorporate with other materials. What is stingless bee honey? Stingless bee honey is made from the nectar of flowers, plant saps, or plant-sucking bug excretions, just like honeybees. There are two main types of stingless bee honey species, which is Melipona and Trigona. And this is the picture of Melipona species, and this is Trigona species. Carbohydrates, water, amino acids, vitamins, and minerals make up a large portion of stingless bee honey. It also contains phenolic acid and flavonoid chemicals. The qualities that makes it suitable for wound dressing product is it is antioxidant, anti-inflammatory, antibacterial, antiviral, anti-hyperlipidemic, anti-diabetic, anti-cancer, and anti-ulcer. Stingless bee honey is available in abundance in tropical and subtropical regions like Malaysia. It has potential medicinal values that is yet to be explored. However, very limited researches have been conducted previously regarding stingless bee honey. Hence, a more systematic research is required to give a complete scientific evidence in order to generate a new therapeutic option that can offer an amazing breakthrough in wound healing applications. Objectives of my research. My first objective is to prepare and characterize the chemical and physical properties of the Gellan gum with stingless bee honey. My second objective is to measure the chemical interaction, mechanical performance, and thermal behavior of the sample. And my last objective is to examine the antibacterial properties of Gellan gum with stingless bee honey. Moving on, the significance of my research is to produce a dressing material from Gellan gum with stingless bee honey as an ideal wound dressing product, and to produce a dressing material which functions to heal the wound via augmentation, pain control, and re-establishment, and to produce a wound dressing product which fulfills all the criteria of excellent wound healing properties to produce an effective wound dressing product. Gellan gum with Manuka honey. Mohammad Azam and Amin conducted a research in 2017 in which they studied the physical and mechanical properties of Gellan gum films incorporated with Manuka honey as wound dressing material. As for the research conducted by Bonifacio and co-researchers in 2018, naturally derived biomaterials were studied in this research due to the lack of it in clinical cartilage repair treatments. Gellan gum in food applications. Moghada Kia and co-researchers conducted a research in which they incorporated red beet extract, or also known as betalains, in Gellan gum together with Salix aegyptiaca distillate to produce gummy candies. As for the research conducted by Rukmani Krishnan and co-researchers, they investigated the use of biopolymers in food packaging as a good alternative instead of synthetic packaging. Gellan gum in tissue engineering applications. In the study conducted by Su and co-researchers in 2018, it demonstrates the tunability of Gellan gum by various chemical modification and incorporation of divalent ions to increase the versatility of the Gellan gum to be applied in variety of biomaterial application. As for another research, which was conducted by Key and co-researchers, the researchers produced a green hydrogel system formulated from microbial polysaccharide, which is salicin and Gellan gum. Moving on to the application of Gellan gum in cell delivery and drug delivery. As for the research conducted by Danka and co-researchers in 2018, they investigated the methotrexate-loaded Gellan gum microparticles for drug delivery. As for the research conducted by Oliveira and co-researchers, they performed the study on enzymatically cross-linked tyramine Gellan gum hydrogels as drug delivery system for rheumatoid arthritis treatment. Wound healing applications of Gellan gum incorporated with oils. In the study conducted by Mukhtar and co-researchers, they investigated accelerated wound healing of physically cross-linked Gellan gum with virgin coconut oil hydrogel containing Manuka honey. As for the research conducted by Mahmood and co-researchers, they evaluated Gellan gum hydrogel films in vitro and in vivo together with the co-impact of therapeutic oils and ofloxacin of wound healing. Wound healing applications of Gellan gum incorporated with nanoparticles. For the study conducted by Ismail and co-researchers, they conducted a study on Gellan gum incorporating titanium dioxide nanoparticles, biofilm as wound dressing, and the physiochemical, mechanical, antibacterial properties and wound healing efficacy were also studied. In the research conducted by Razali and co-researchers, they studied the titanium dioxide nanotubes incorporated Gellan gum bio-nanocomposite film for wound healing and the effects of titanium dioxide nanotubes concentration. The materials needed: high acyl Gellan gum or chalcogel, Mueller-Hinton-Diffco agar powder, stingless bee honey from the species of Trigona itama, and anhydrous calcium chloride. The apparatus needed will be beaker, glass rod, cotton swab, sterile petri dish, laboratory scales, autoclave Perkin-Elmer spectrum, Pipe Miracle ATR accessory, Instron universal testing machine, Biomerix DensiCheck. The Gellan gum films were prepared via casting method. The first step is the Gellan gum solution was prepared by dissolving 1% of Gellan gum in 70 ml deionized water and with 55% glycerin at continuous stirring for 1 hour and 45 minutes at 70 degrees Celsius, followed by, after the Gellan gum was fully dissolved, 5 ml of calcium chloride was added into the mixture and stirred again. Lastly, Manuka honey was added and the solution was stirred for another 15 minutes. These Gellan gum solutions containing 2%, 4%, 6%, 8%, and 10% Manuka honey were known as Gel H2, Gel H4, H6, H8, and H10 respectively. The solution was poured into petri dish and dried in the oven at 50 degrees Celsius for 24 hours. The characterization that will be performed is FTIR characterization, swelling percentage, gel fraction, water vapor transmission rates, tensile strength, and antibacterial activity. As for the expected results, I would expect the successful incorporation of stingless bee honey into the Gellan gum film. The swelling percentage is expected to decrease with the addition of honey. Shifting and disappearance of the frequency in the functional group peaks are expected in the FTIR spectrum. The sample with the presence of honey is expected to show effective antibacterial activity against gram-positive and gram-negative bacteria. The sample with inclusion of honey is expected to have low tensile strength. The sample with lowest percentage of honey is expected to show high water vapor transition rates value compared to the sample with higher percentage of honey. And finally, the sample with the highest percentage of honey is expected to have the lowest gel fraction. With that, I thank you, Doctor.